And now we will hear Katharina from Berlin. So on the program you see that normally it's up to Volker, which is the group leader of where Katharina is working, but he couldn't manage to come. So now Katharina will make a merge of both talks and present the work that she's doing at the Ph.D. student in Berlin. Thank you, Katharina.
Ja, guten Tag alle zusammen. Vielen Dank für die Einladung.
Hi everybody, and thank you for inviting me to this conference. The key question that Volker and my work is concerned with is what role do lipids play in the cell? Very important is the myotubularin, MTM1. This is one of the genes that can have an impact on the structure of cells. This is a picture of a cell, and this cell is split up in different compartments. In order to communicate, so to say, with other cells and exchange information, exchange matter, in order to do that, the cell has to be able to process these things like hormones or proteins. This process is called exocytosis. And is comprised of endosomes, which are transport vesicles, and these endosomes pass on the information of hormones and proteins to the cell. To give information away from the cell, the process is called exocytosis, and does the same but in reverse. These compartments work similar to an apartment. They are separated by fixed walls, and each compartment has a specific role. Some of the compartments are essential for taking in energy. Others are there to clean up the cell, and these different compartments in the cell are separated by membranes. This is how inside a cell there can be different processes taking place. These membranes inside a cell are made up by lipids and clearly separate one function from the other. These lipids are consistent of a water-repellent tail. You can see it by this green zone on this picture, and it is hydrophilic. A second part of the lipids are the water-attracting lipids. These are filled with water. That means that there are compartments separated from one another by water. And that means that the compartments above and below can have different functions. There are different types of lipids in cells. There are structural lipids that define how the membranes are formed, if they are round or straight, if they are rough or smooth. And those are the structural lipids. What we are concerned with are the phosphonosoid lipids, the PIs. And they give more information about the cell, what the name of the cell is, what its function is. And those identity lipids, identity-giving lipids, are set up in circular structures. They can be added on in position 2 to 6. There can be different PIs attached. And depending on where the phosphate groups are, a new compartment is defined. And these lipids define the identity of the compartments. They decide, is it the bedroom, is it the kitchen, is it the bathroom of the cell. If you look at a cell, this is another picture. These intracellular compartments are seen here and are colored differently. They all have a different code, so every compartment has a different identity defined by its lipids. If you want to work with this cell, you have to change the code of the cell. For example, if I want to change clothes going from one room to the other, I adapt my identity to a new social setting. For example, if I go to work, if I go to sports. And we have to imagine it the same way within the cell. The compartments, if they want to be transported, have to change identity. The surface is defined by phosphates, PI4P, and on the right-hand side, the phosphates are mostly on position 3. We basically exchange identity coming from outside of the cell to the inside and vice versa. How is it possible that a compartment changes its identity? Well, the compartments remain the same, but they change their appearance. Imagine it like stepping into a river. You step into a river at one place and leave at another place. It is the same river, but again, it's not the same river because it's not the same water that surrounds you. This is the analogy that we use, the difference between remaining and changing identity. This is another simplified picture of a cell. We have the surface with the PI group 4. When we go from outside to inside, it is very easy to understand the way of transport. The 4 phosphates are split up by enzymes. Then we have the 3 kinases. This is how we change the identity of from PI4 to PI3. We have seen from transporting information from inside to outside, we have found that Myotubularin 1 changes PI3P to a simple PI. We have found out that myotubularin is sitting at this very compartment and essentially influences the transport of information to the outside of the cell. This is particularly important for integrins inside the muscle. These integrins are structural elements of the muscular cell. We have talked about that muscles need a structure to make use of their function. These integrins are usually sitting towards the surface of the cell and are essential to myotubes, to develop. And if this isn't possible, then the integrins remain inside of the muscle cell. Myotubularin is not the sole enzyme of these PI groups that change lipids and fats. There are many of these phosphatases that are different in structure. However, all of the myopathies have some sort of involvement with these PIs. We have focused on the PI3 group, but there is a lot of research that needs to be done in the other areas as well. What I wanted to talk about in a bit more detail is how does myotubularin work together with lipids, and how is it influencing intracellular transport? How is this possible to do with fats? You can fill up the transport paths with a staining element. These staining colors are taken in by these transport cells. They are grouped in the endosome zone and then transported out to the surface. This can be looked at by integrins, but also it's the same procedure for other proteins. This is a control cell where we're just looking at the enriched staining that is here illustrated with green dots and that is transported to the outside. What we will see in the film is how transport vessels are seen at the surface of the cell and then all of a sudden disappear. And this expression is characterized by this abrupt change. If you see those little circles, you see that these events take place and then suddenly disappear, and that they happen very repeatedly within the cells of healthy human beings. The contrast in this room may not be perfect, but I hope you can still see the dots. If you look at the cells of CNM patients, people who are lacking MTM1, then you can see that these structures are enriched, that they are quite, that they are not as mobile, and that these staining elements remain inside of the cell, cannot be transported out, and that transport path is not working. That means that the integrin that is important for muscular structure cannot be transported to the outside, and muscular cells cannot properly develop, and these integrins remain in the inside of the cell. That means that muscular fibers are not fully developed. What can be done is, if you look at myotubularin that is sitting on endosomes, which is a substrate of myotubularin. At the same time, there is the PI3 kinase, and all these enzymes work in equilibrium. Myotubularin, PI3, and PI work together, and the cell needs a working equilibrium of kinases, myotubularin or PI3. If there is little myotubularin, then PI3 is enriched. What we have tried to do is, if there is a cell with a lot of myotubularin, we attempted to switch off the PI3 kinase. So that situation that is often taking place in CNM patients inhibits 3 kinase PI3. If we take the cells from the patients and we work on them as I've just described, then they become control cells. They are set back to the original state. It is therefore possible to enable the transport inside the cell. Other things that we have done is we have looked at integrins. Those are the red dots, This is a patient's cell, and the red dots are not present in normal cells. These enriched elements are transport vessels. Because the transport is inhibited, there are more and more of these integrins. Now, if the inhibitor of PI3 kinase is applied, then all of a sudden the accumulation of integrin can be stopped and can be helped. Here we see an accumulation of the integrins. Then the inhibitor, is applied, and over time there is a reduction of myotubularin. In these patient cells, you can see that possibly by giving more of the inhibitor, the problem can be mended. To sum this up again, we see MTM1 sitting on top. This, together with the kinases, regulate the PI4. We have found a second enzyme, and that works together with the PI3 that loses its identity. And then we add a phosphate group coming to PI4, which enables the communication to the outside of the cell. This regulates the transport or the export of integrins, of structural components, from the inside of the cell to the outside of the cell. We expect that this faulty transport to the outside of the cell is partly responsible for structural issues inside of the cell, and that the PI3 kinase can partly mend this fault. We are not sure of how this can practically be tested. I would like to thank everybody who was involved. My boss, Volker Hauke, Mr. Schulz, Jocelyn Laporte, and Anne-Sophie Nicot, of course, who have given us the patient cells. Thank you very much.
Okay, vielen Dank. Irgendwelche Fragen an Katharina? Also wenn ich das
Any questions to Katharina? Oh, Holger, you've got one. If I understood you correctly, this inhibitor is improving the situation in the muscle cells, and I assume you tested that in individual cells now only. So my question is, what kind of substance is it? How far is it advanced in order to become a medication? Has it been tested in vivo or in animals? Not now, no, not yet. We had patient cells, that was muscle cells, fibroblast cells, which was skin cells, and we did cell culture experiments. So we looked at individual cells, where when you add the inhibitor over 1 or 2 weeks, these cells have a better survival, so they can better kind of degrade the aggregates, this enrichment of substances in the cell. But these are just experiments over 1 or 2 weeks. And to what extent this then can be applied in human beings, that's a very small pharmacological substance still. So we haven't got yet mouse experiments. It might be interesting to test it in mice, actually. However, we don't have mice ourselves, so we can't. We do have mice. But we're working together with you. May I summarize this? Katharina is really doing foundational research. And when I'm correct, the myotubularin has a certain role in a cell, and it's part of a network. And this myotubularin has a certain network, and there are other proteins who are counter agents, so to speak. And what Katharina has found out, that when the myotubularin doesn't work, one could try actually to block the counter protein. And that worked rather well in the cells, but as I said, this is still basic research. Katharina hasn't yet worked with mice. That would be the next goal, of course, to test this inhibitor of the counter protein of myotubularin. But this is pharmacological things now. What she does is basic research. This is just foundations. And even for professionals, it's sometimes very difficult to understand, but Katharina is a real expert in this field.
Macht ja nichts. Haben wir wenigstens alles was gelernt, ne?
Yeah, but we learned something. One more question over here. Regarding the basics. How specific is that system for muscle cells? Because it will be different if you interfere with such a basic principle that you find in any cell. How tolerable will it be in the end if I develop a substance out of it? And I think this is the most important issue. Well, the mechanism that we detected is universal, so it works in all kinds of cells. MTM1 is not a muscular-specific substance. This enzyme is present in all other cells as well with a certain function. So I suppose if you do inhibitor experiments, you have to look at the dosing. Depending on to what extent you inhibit the counter enzyme, you should never delete it completely. That wouldn't be healthy, I think. But the matter is the dose, I suppose. And as far as I know, in patients with the longer survivals, other organs would be affected as well, not just muscle cells. So MTN1 will have a function in other organs as well. The muscle cells will probably be only the first ones that suffer from not having MTM. So I think I don't really know whether this is only muscle specific, whether it's just that the disease first presents itself in the muscle cells. Is that answering your question? I mean, you can clarify that in more detail later on during the coffee break, can't you?